CAR T-cell therapy, a living drug. As their name implies, T-cells, which help orchestrate the immune response and directly kill cells infected by pathogens, are the backbone of CAR T-cell therapy. Currently available CAR T-cell therapies are customized for each individual patient. They are made by collecting T-cells from the patient and re-engineering them in the laboratory to produce proteins on their surface called chimeric antigen receptors, or CARs. The CARs recognize and bind to specific proteins, or antigens, on the surface of cancer cells. These receptors are synthetic molecules, they don't exist naturally, explained Carl June, MD, of the University of Pennsylvania Abramson Cancer Center, another leader in the cellular therapy field. After the revamped T-cells are expanded into the millions in the laboratory, they're then infused back into the patient. If all goes as planned, the CAR T-cells will continue to multiply in the patient's body and, with guidance from their engineered receptor, recognize and kill any cancer cells that harbor the target antigen on their surfaces. The CAR T-cell therapies approved by FDA to date target one of two antigens on B cells, CD19 or BCMA. Making a CAR T-cell. A growing number of CAR T-cell therapies are being developed and tested in clinical studies. Although there are important differences between each specific therapy that can affect how they function in patients, they all share similar components. Each CAR bridges the cell membrane. Part of the receptor is located outside the cell and part within the cell. The part of the CAR that extends out from the cell's surface is usually composed of fragments, or domains, of lab-made antibodies. Which domains are used affects how well the receptor recognizes or binds to the antigen on tumor cells. The internal part of each CAR has signaling and costimulatory domains. These transmit signals into the cell after the receptor interacts with an antigen. The different domains that are used can affect the cell's overall function. New treatment options where few existed. The initial development of CAR T-cell therapies focused largely on the most common cancer in children, acute lymphoblastic leukemia, ALL. More than 80% of children diagnosed with ALL that arises in B cells, the predominant type of pediatric ALL, will be cured by intensive chemotherapy. But effective treatments have been limited for patients whose cancers return or relapse after chemotherapy or a stem cell transplant. Second CAR T-cell therapy approved for lymphoma tisagenlecleucel is a new option for some patients with common lymphoma. In 2017, however, a new option appeared, with FDA's approval of tisagenlecleucel, Kymriah, the first CAR T-cell therapy to be approved by the agency, based on clinical trials demonstrating it could eradicate cancer in children with relapsed ALL. In some cases, CAR T-cell therapy has now been studied long enough that details about the long-term outcomes in children are beginning to emerge. An NCI-led research team, for example, recently reported on long-term follow-up from children with relapsed ALL who had been treated with CAR T-cells as part of a clinical trial. More than half the children went on to receive a potentially curative stem cell transplant, they found. And approximately 60% of those children were still alive 5 years later without their cancer coming back or the children experiencing any disease-related problems. The progress made with CAR T-cell therapy in children with ALL has been fantastic, said Terry Fry, MD, who has led several clinical trials of CAR T-cell therapies at NCI and, more recently, at Children's Hospital Colorado. As CAR T-cell therapy has become more widely available, Dr. Fry said, it has rapidly become the standard of care for children with relapsed ALL. CD19 targeted CAR T-cells are also offering hope to adults and children with advanced aggressive lymphomas. Before the development of CAR T-cells, many of these patients were virtually untreatable, said James Kochenderfer, MD, of NCI Center for Cancer Research, who has led several trials of CAR T-cell therapies in patients with diffuse large B-cell lymphoma. The results in lymphoma to date have been incredibly successful, Dr. Kochenderfer said, and CAR T-cells have become a frequently used therapy for several types of lymphoma. Understanding and managing the side effects of CAR T-cell therapies. Like all cancer treatments, CAR T-cell therapies can cause severe side effects, including a mass die-off of antibody-producing B cells and infections. One of the most frequent and serious side effects is cytokine release syndrome, CRS. As part of their immune-related duties, T cells release cytokines, chemical messengers that help stimulate and direct the immune response. In the case of CRS, the infused T cells flood the bloodstream with cytokines, causing serious side effects, including dangerously high fevers and precipitous drops in blood pressure. In some cases, severe CRS can be fatal. Ironically, CRS is considered an on-target effect of CAR T-cell therapy, that is, its presence demonstrates that T cells are at work in the body. Generally, patients with the most extensive amount of cancer in their bodies are more likely to experience severe CRS from CAR T cells, Dr. Kochenderfer explained. In many patients, both children and adults, mild forms of CRS can be managed with standard supportive therapies, including steroids. And as researchers have gained more experience with CAR T-cell therapy, they've discovered ways to better manage the more serious cases of CRS. A big part of that management is the drug tocilizumab, Actemra. This drug, initially used to treat inflammatory conditions like juvenile arthritis, blocks the activity of IL-6, a cytokine that is often secreted in large amounts by T cells and macrophages. The other side effect of particular concern with CAR T-cell therapies is neurologic effects, including severe confusion, seizure-like activity, and impaired speech. The precise cause of these neurologic side effects, also called immune effector cell-associated neurotoxicity syndrome, or ICANS, is still unclear. Although it's effective for treating CRS, tocilizumab doesn't seem to help with ICANS. Steroids are currently the best treatment option for severe ICANS, particularly dexamethasone, which is better at penetrating the central nervous system than other steroids, explained Jennifer Brudno, MD, who is involved in several trials of CAR T-cell therapies in NCI's Center for Cancer Research. A diagram of a CAR T-cell highlighting the changed components of the engineered receptor. Remodeled CAR T-cell therapy causes fewer side effects for people with lymphoma. Treatment safer, as effective as original CAR T-cell design. Different ways of preventing CRS and ICANS are now under intense study, Dr. Brudno said. One approach is the prophylactic use of tocilizumab and low-dose steroids. Although further studies are needed, the data so far are reassuring, she said. Other treatments for ICANS are being investigated. Smaller studies, for example, have found that Anakinra, Kineret, which is used to treat rheumatoid arthritis, may help prevent severe ICANS in patients treated with CAR T-cell therapies. Exit disclaimer. One more idea for protecting against severe CRS and ICANS is to modify the CARs themselves, Dr. Brudno explained. In a small clinical trial of adults with lymphoma, for example, patients treated with a remodeled CD19-targeted CAR T-cell developed at NCI had far fewer severe neurologic side effects than historically seen with the original form of the same CAR.